Hello, my name is Evan Clark, and today I'm going to be talking about a process called two-photon upconversion. Two-photon upconversion is a process by which two photons of a lower energy are absorbed by a molecule or a system of molecules and are converted into a single photon of a higher energy. Now, this should be distinguished from two-photon absorption, where two photons are absorbed simultaneously. In this process, two photons are absorbed sequentially. Additionally, in this process, there are a number of energy transfer steps to get the energy from those two original low-energy photons to be combined into one single higher-energy photon. Quickly, we will talk about regular fluorescence. So, in a typical system, incoming light of sufficient energy will excite an electron from the ground state to an excited state. Through a number of different possible mechanisms, some of this energy can be lost. One of these methods is two different vibrational states relaxing, but none of these processes release energy as light. They typically release them as heat or motion. After some time, this electron will then fall back down to the ground state and will release this energy in an emitted photon. Because this second difference in energy between the excited state and ground state is shorter than the original difference in energy levels, the emitted light is going to have a longer wavelength and lower energy than the incident light beam. But for any particular molecular system, this emitted light will have a characteristic frequency that it will emit. This shift of wavelength of light from absorption to emission is what we call a Stokes shift, and this is the typical kind of shift in frequency or wavelength that we would expect to see when we're looking at something that is fluorescent. What we can see in this image is that we have two different incident light beams. On the top we have a green laser, and on the bottom we have an ultraviolet laser. On the solution on the left, both the green and the ultraviolet lasers give sufficient energy to promote electrons to an excited state and allow the solution on the left to fluoresce yellow light. However, what we see on the right, our ultraviolet laser light does have enough energy to promote electrons to an excited state, allow them to radiate some of that energy off into vibrational motion, and then when the electron falls back down, we can emit blue light. But the green laser does not provide sufficient energy to promote an electron from the ground state to the excited state in the solution on the right. Since this molecule emits blue light, which is of higher energy than green light, in a simple one-photon process, this would go against the first law of thermodynamics, to have a single photon go from low energy to high energy without any other source of energy input. We can see this again in this video clip. Under ultraviolet light, all of these samples glow their respective fluorescence colors because ultraviolet light has enough energy to cause all of these samples to fluoresce. But if we switch to a lower energy blue light, now the sample on our left does not have enough energy to fluoresce. It does not absorb nor emit blue light. If we decrease the energy of the incoming light again to green, then we see that the two samples on the left no longer are able to fluoresce. The incoming light does not have sufficient energy, and since they do not absorb or emit any light, they look transparent, while the two samples on the right still do fluoresce because the green light is enough to excite their electrons. Again, if we decrease the energy of light to yellow, we see that now the three samples on the left all appear to be transparent because they can't fluoresce, while the one furthest to the right still does fluoresce red because the yellow light has enough energy. If we decrease the energy of light one last time to red light, we now see that all four of these samples are unable to fluoresce because the red light does not have sufficient energy to excite the electrons in any of these samples, so all of these samples appear transparent. Another important thing to note is the definition of a singlet state and a triplet state. So in a singlet ground state, we have all of our electrons on the lowest energy and their spins are paired. If we excite the electrons into a singlet excited state, then we retain the original alignment of the spins, but one of the electrons has been promoted to a higher energy state. This can then relax into a triplet excited state where we have our spins unpaired, and one of them is in an excited state. Now let's talk about the mechanism briefly for two-photon upconversion. The mechanism we'll be talking about is triplet-triplet annihilation. From the beginning, what we have is two low-energy photons absorbed by a sensitizer molecule. These molecules are important because they can easily absorb low-energy light, such as red light or infrared light. They are typically polycyclic aromatic hydrocarbons or other molecules that have large conjugated systems that are easy to excite. The excitation from these sensitizer molecules is transferred to emitter molecules. This relaxes the sensitizer molecule back to its ground state and brings the emitter molecules into an excited state. Now, these two emitter molecules are in a triplet state, and we will see how that happens in just a moment, but these two triplet states can annihilate, bringing one of these two emitter molecules back to the ground state and promoting the other one to an even higher excited singlet state. And this then undergoes an electronic transition which emits a blue photon of light. So we can see in summary that we have two low-energy photons being converted through a series of electronic transfers into a single higher-energy photon. And this is cool because in one photon processes, we said that this wasn't really possible. We have increased the energy of our photons, and we did this by combining the energy of two photons. So, whereas in a normal system where the frequency of light decreases after emission, and we call that a Stokes shift, we call this kind of shift an anti-Stokes shift. Now, getting into a little bit more detail, what we see is on our sensitizer molecule, we absorb a photon, and that promotes it from a ground state singlet state to an excited singlet state. Through some intersystem crossing, where we lose energy to our vibrational modes, this is brought down to a triplet state. This triplet energy can be transferred to the emitter molecule, again bringing this electron down to its ground state in the sensitizer molecule and promoting a ground state electron in the emitter molecule into a triplet state. This happens a second time with another pair of sensitizer and emitter molecules, so we have two emitter molecules in a triplet state. These two molecules can then interact, and it brings one of their triplet states back down to a singlet state, and it promotes the other one up to an excited singlet state. We have effectively transferred the energy of these two low-energy photons into a high-energy state on a single emitter molecule. When this fluoresces, we then release that higher energy photon. This technique was first applied to microscopy in 1990. This team used two-photon laser scanning fluorescence microscopy to image chromosomes in cells. They stained the DNA with fluorescent molecules, and using this two-photon absorption technique, could image these chromosomes. One of the more recent developments in applications for this type of two-photon upconversion is in photovoltaic cells for solar panels. So without getting into too much detail on how solar panels work, basically you need light with sufficient enough energy to make your solar panel work. And this, as we can see in the diagram, it works just fine with blue, green, yellow, and orange light, but with more red light and with infrared light, which is a significant amount of solar radiation, this is all unusable energy, and it goes wasted. We can increase the efficiency of our solar panels by converting this red and infrared light into higher energy light, such as yellow light seen here. The red and infrared light pass through the solar cell, then interacts with our triplet-triplet annihilation solution, and that releases from that solution yellow light or higher energy light, which can then interact with our solar cell and produce more electricity than would have been previously possible. Now, some of the benefits of using this kind of fluorescence for microscopy is that it allows for deeper penetration into tissue samples up to 500 micrometers, and it uses lower energy light to do so, and so you have less risk of damaging your samples with higher energy light. Most traditional fluorescence microscopy exhibits pretty poor tissue penetration at the excitation range, so at ultraviolets or lower wavelength visible light. This necessitates the need for even higher power in the irradiation, which results in significantly more noise in your background image due to background fluorescence, and it increases the risk of damaging your sample even more. Some of the limitations of this are, though, that molecular dioxygen is, in its ground state, a triplet state. And because of this, it is able to quench the triplet states in the chromophores in a non-radiative manner. So this decreases the efficiency of your samples, and it also produces singlet oxygen. The other main drawback of this is that these chromophores, since they are usually large conjugated systems, are not typically very soluble in aqueous solvents, and sometimes that solubility problem can become an issue. But as we'll see in a case study in a few minutes, there are ways around this. So in a setup that is pretty typical for fluorescence, we have a few different ways of setting up our excitation beams and detector lenses. But most of them consist of an incident light beam reflecting off of a mirror through an objective lens onto or through the sample. The sample then fluoresces, and that emission then passes through the objective lens again and passes through this two-way mirror. That light is then detected by a monochromator and a detector, and you can get some really nice imaging data of your samples this way. In this case study, we will look at a system where a triplet-triplet annihilation system is encapsulated in a silica-based nanocapsule, or micelle, which is able to be functionalized on the surface to allow for discrimination between different targets. This also ensures that the sensitizer molecules and the emitter molecules, or acceptor molecules, are in close proximity to each other in higher concentration. Being enclosed in a micelle helps disperse them in a solution when otherwise they would be completely insoluble, and the oleic acid prevents triplet oxygen from quenching the reaction. Here, two different SNCs were synthesized, one with an emitter light that emits blue light, and one with an emitter that emits green light. As we can see here on either side on the bottom, with these two systems, just the oleic acid, which forms our micelles, does not fluoresce. A solution of the sensitizer molecules and the acceptor molecules isolated in the oleic acid does not fluoresce. Only when the sensitizer and emitter molecules are able to interact with each other in solution do we get fluorescence. So with one we get blue fluorescence, and in the other we get green fluorescence. Here we see the formation of these silica nanocapsules. First, oleic acid is used not only as a solvent to dissolve these chromophores, but also as the surfactant to form the micelles and as the quencher for ground state triplet oxygen. The chromophores were then dissolved in the oleic acid and sonicated with vigorous mixing, and then with the addition of 3-aminopropyl triethylsilane, the micelles formed. These micelles were then further reacted with tetraethyl orthosilicate to form silica shells. The surfaces of these nanocapsules were then functionalized with amino groups by adding 3-aminopropyl trimethoxysilane. This allows for the covalent attachment of cancer-specific bioprobes, such as antibodies or peptide sequences. By functionalizing the blue fluorescing silica nanocapsules with antibodies and functionalizing the green fluorescing silica nanocapsules with peptides, this allows for the nanocapsules to target specific cancer cells and emit color-specific fluorescence based on the type of cancer that it is. And it can do this with one specific incident light of low energy. And as we can see with this detector setup, when 635 nanometer red light passes into the sample, it then fluoresces out either 475 nanometer blue light, or 505 nanometer green light, depending on the type of cancer and the type of silica nanocapsules in the cells. This light is then detected and then processed so that we can get images of these specific cells. To demonstrate this application, this was performed in an in vivo setting. A custom-built upconversion imaging system, as depicted here, was constructed, allowing for the imaging of animal specimen. Both of these silica nanocapsules were injected into the tail of the mouse and allowed to accumulate in the body. And as you can see in the image, these targeted the specific tumor sites very well. When these peptide and antibody targeting moieties were not incorporated onto the silica nanoparticles, there was some accumulation seen due to the EPR effect, but there was a significantly enhanced imaging ability when these targeting moieties were present. So, in summary, two-photon upconversion is a process by which sequentially absorbed photons of low energy lead to the emission of a single photon of higher energy. This is done through the transfer of energy between sensitizer and emitter molecules and through a triplet-triplet annihilation. This atypical shift of wavelength to shorter wavelengths is known as an anti-Stokes shift, and has many promising potential applications in solar panels and photovoltaic cells, as well as biomedical imaging and fluorescence spectroscopy. Thank you.